now. Thank you, Joel. And I think that, yeah, now I'm recording the video. Yeah, so those, Winifred, are the three standard parameters, I would say, that are accepted. So mineral frequency, the reason to use a mineral frequency of 5% is that if you use a lower number than that, it's gonna be so difficult to identify. If you are using GWAS, for example, it's gonna be so difficult to identify um, an association with an allele that is in just 1% of your population, for example. It's gonna be tricky. So that is why usually we use 5%, but there are some people that think that that's a mistake, because as breeder, maybe we are interested in really rare variants in the genome that can cause, for example, resistance for some disease. So that depend of your objective and depend of your of your project. But the standard is just filtered by 5%. Okay. And another question that have Winifred, it's about the second paper of the second video, when they mentioned that they basically, and I send you that video because I want you to think that if you have, for example, if the population that perturbed for Harding-Weinberg is the most probable reason is because a natural variation, or because you have subpopulations or selection, that is what normally happen. But if you are working with sequencing data, and as if you are interested in genomic selection, you are going to work with sequencing data. One alternative way to have a population that is not in Harding-Weinberg equilibrium, or some factor that can influence your data analysis, is that you can get errors in the genotyping. And that is common. And if you are using methodologies as GPS that are kind of high throughput genotyping methods, you are going to have genotyping errors. And we are not going to talk about that in the hands-on session, Winnie. But what I want to share you here, this slide, is that we can try to control, or in some degree, we can control that genotyping error again with the quality. So if we filter all the sequencing data or the SNP data with a quality of 40, with that value, I think that in some degree you can be sure that the nucleotide that you have, or the SNP that you have, it's good enough, and it's not a mistake from the genotyping methodology. Okay. So I think that we have some questions here in the chat. I'm going to try to read them. Okay, so Symphorine asked me about, in case someone is working in an orphan crop where there is no reference genome available, what is advisable? Well, methodologies as GBS are so popular right now because you don't need a reference genome. So because in GBS, basically you just have to cut the genome, the DNA with a restriction enzyme, and then you can put together all those pieces of the sample, and basically you and put all the pieces of your sample and the population, and you can identify the variations, the SNP variation, or the molecular markers in your population without use a reference genome. So that is possible, and there are multiple tools for to do that. I'm gonna send you, if you please send me a message in Slack. I'm gonna share with you some one paper that actually I use the pipeline, or the software. The name of the software is Slack, and is Stack, and basically what Stack does is try to put together all the information that seems that is similar, and in that way you can identify the molecular marker. So I did that in CowP a couple of years ago, and that works. Obviously, it's better when you have a reference genome because you have all the genes identified in that reference genome, and it's just gonna be easier. But now with methodologies as GBS, it is possible to advance as well. And the good thing with GBS is that in theory, it's cheap, or it's relatively cheap. So you can do that. Let me see if there are more questions here. I can share this presentation if you want. Yeah, I can share that. There are not a lot of information here, but if you want the presentation, I can share that with you. If you have questions, I am trying to read the chat, but if you have questions, please open your microphone, and maybe that is faster. And again, I want to keep that like an informal conversation, so don't worry about that. Yeah, just open and we can talk between us.
I have a question, Paulo.
Great, go ahead.
Thank you. On the first video on molecular basis of genetics, around the 4 minutes and the 48 seconds, we said when doing genomic selection, we select individuals with beta genetic values and cross them by keeping the genetic diversity. Can you please elaborate more on the genetic diversity kept?
Okay, but okay, yeah, great for the question. So I didn't want to go to that genomic selection thing because that we're gonna discuss that at the end of the third week. But as you open your microphone, so I'm gonna answer that. So the idea is that with genomic selection, we can estimate the genetic value of the lines. Yep. And basically that genetic value is like a representation that we have to basically say, this is the genetic component that explains the phenotype that we're interested on. So if we have a high genetic value for a specific trait, so that means that in theory, at least with the genetic portion that we can explain that phenotype, we predict to have a high phenotype as well. So basically that it's how we can explain the variability in the phenotype with the genotype. And when I talk about how we can increase the genetic diversity in the germplasm, is because usually what happens in plant breeding is that we have the top lines that have the best yield or seed production. Sorry, and we try to cross between them, so we can we tend to cross the best line with the best lines, which is it's good. But the problem with that is that in some cases there are some of those lines that are good have a really strong pedigree relationship. And that is bad. That is good to short term because you are gonna just cross the best with the best. But the problem is that the diversity each time that you cross lines that have a strong relationship with the pedigree is gonna be lower and lower. And at the end, in the short term, you are not gonna have a lot of diversity to use in your breeding program. So with the genetic value, you can estimate lines that have a good genetic value, but at the same time, as you have the molecular markers, we can cross the lines that have good genetic value, but have more diversity between them. So basically with that, you can try to have a genetic gain to the long term in the breeding. Yeah, because you are not just gonna cross the best with the best, but you are gonna cross the lines that are best for the phenotype that you are interested on, but also you are gonna take into account the diversity between those lines. And that is something that we are gonna talk later. But that is a really good point, and that is one way to have genomic selection to try to help to the genetic gain in the long term in the breeding programs. Did I answer your question, Tembo? Uh,
Hello.
yep.
No, I have a question.
Go ahead.
Yeah, it's called Immaculate from Massey, Macquarie University. I was concerning heritability. I saw the presentation on heritability, broad sense and narrow sense. I'm wondering what you'd advise if I'm working on a trait that before the heritability most times is kind of like really low. What would you advise if you're to relate it to the broad sense and narrow sense heritability? Because in your presentation, you said narrow sense heritability is more important to breeders. Now I'm trying to relate it to a trait that has, usually they find that the heritability is quite low. I'd like to hear what you have to say concerning that. How can it be handled or improved? Thank you.
But I didn't understand your question. Could you repeat? I understand that you are asking me about the heritability and how we can use in the breeding program. But
okay, I was saying, I got it clear from your presentation that narrow sense heritability is more important than the broad sense. So I'm kind of trying to relate it to a trait that I'm working on that usually previous researchers have always found that the heritability values are quite low. What would you advise when someone is handling research on that concerning the broad sense and the narrow sense?
okay. Yeah. I just are you seeing my screen, or are you like, could you watch my screen or my presenter view? So because somebody just told me that they okay, great. Yeah, you can not separate between both heritability, but the concepts are there. So let me show you one slide that we have, I think, here. Hopefully it's here. Okay, so the thing is that the broad sense heritability, it's all the genetic component that you have in that trait, and that include dominance, that include epistasis, and you can it's kind of complex, and you can basically evaluate the broad sense heritability of in common being, because we have pure lines. So if you planted the lines in different, the genotype that is pure in different environments, you are gonna see what is the heritability of that plant, because like it's basically a clone, so all the genotype is gonna be the same because it's a pure line. So you can basically estimate what is the dominance deviation, what is the epistasis effects that have that genotype with the environment, because Yeah, basically remember that in both heritabilities, but in the broad sense heritability, you take into account the genetic variability variance over the phenotypic variance, and the phenotypic variance take into account the genotypic variance and the phenotypic variance. So basically, there are like an environmental component there. And that it's good to understand how pure lines or clones, what is the performance of those pure lines or clones in different environments, because that is the only way, with pure lines is the only way that you can keep like all those variations in the same individual. But when I mentioned that narrow sense heritability is most important, is because we are interested to produce more populations. We are interested to produce a genetic variation. And broad sense and narrow sense heritability just try to take into account the additive effect of the alleles. And with additive effect, basically mention, or what I want to explain is that they have in diploid individual, they have two alleles. So we want to estimate, we want to give them, give to the genotype a value. First, we have to estimate the value or the effect of one of those, or of both alleles. And then we want to estimate or give to the genotype that have a combination of those two both alleles value. And that it's going to be the performance that that genotype is going to have in an additive effect if all the gene has an additive effect. But also it's going to be the performance, where we can estimate the performance that the population, the offspring of that genotype is going to have on average. So that is why in the breeding perspective, additive values or additive, effects and narrow sense heritability, it's important, because that it's the heritability that is transmissible to the next generation. If you have a low heritability, and always the narrow sense is going to be a little lower than the broad sense heritability. And there are just traits that have a low heritability. Some of the most important, for example, seed production, always have a low heritability, and that is because, or that, it's because there are a lot of regions in the genome that have association with those phenotypes. And when there are more regions that have association, the effect of all the markers, the effect of those regions is smaller and smaller. And again, if there are more regions in the genome, there are more regions also that are going to have an interaction with the environments and or with external factors. So that it's why there are some traits that the heritability is low, and the only way that you can try to increase that heritability is just to control your experiments and try to reduce the error as much as possible. Something that I am using to try to increase the heritability, for example, in seed production, is using a spatial analysis and try to take into account in the environments that I am working on the distribution of my plots, because there are some areas in the field that have, for example, more water or more nutrients, and that can have an impact in the seed production. So when you take into account the spatial distribution of your experiments, you can increase a little bit your heritability as well. So basically reducing the errors can help that. Yeah, and I was clear with that, or do you have more questions about that?
No, thank you. I think it's clear now.
Okay. Yeah, but the important thing is that again, the broad sense take into account not just the additive effect, but dominance and epistasis. And the additive one, it's as breeder again most important because we can predict the phenotype in the progeny of those individuals because yeah, the effect is additive.
I still have a question on that.
Yeah, great. Go ahead.
Thank you. Next slide to that.
I don't have all the slides here. I just select some of them, but let me check. This one, no.
Yes, this one.
Right.
Yes, this one. We can see that from this, there is a graph where the offspring height is regressed against mid-parent height, right?
Mm-hmm.
And the slope is 0.84. I guess that is the narrow sense heritability, right?
Yep.
Yes. So now, does it mean that does it mean to get the narrow sense heritability is just to regress the offspring value against the mid-parent value only, or there are some other explanatory or independent variables that have to be included?
That is a wonderful question, and this is the simplest scenario. So, and yeah, you are right. If you have the mid-parent value, the slope of that regression, if you regress the offspring, to the mid-parent value, the slope, it's gonna be equal to the narrow sense heritability, and that makes sense because as you in an additive model, the offspring should to be in the middle of both parents, as we have here in this graphic. So we have, for example, the homozygous B1 and the homozygous B2, and the offspring of that cross should be in the middle. So that is why we have to estimate the mid-parent value that is going to be in the midpoint between both homozygous. And if the regression is perfect, that means that there is not environmental variables that influence that effect. But the regression is not perfect, like this case that it's pretty good, it's 0.8, but it's there. We have some points that are around the line that there is the regression. So that means that there are some environmental effects. And you can do other kind of regressions. For example, if you just have the value of one of the parents, you can do also the linear regression between the offspring and one parent. But in that scenario, the narrow sense heritability is not going to be equal to the slope. It's going to be equal to the half of the slope because you are just using the information of one of the parents. And there are some of those variations. And that is a pretty simple concept. I hope so. But there are more complex ways to estimate the additive relationship. And one of them is with molecular mark when we have dozens of millions of molecular markers and we don't have the information of the pedigree information between them. So one way to that we can use to estimate that additive relationship between the lines, it's take into account all the markers that are similar. I think that maybe we can discuss that later because that is kind of complex. But yeah, we can use also the molecular markers to estimate the heritability of the trait, the narrow sense heritability of the trait. Basically, we try to measure the relationship at sequence level between the lines. And in that way, we can estimate the heritability, the narrow sense heritability. Because remember, narrow sense heritability is how basically how many genes or regions they share between the lines. So as much genomic regions they share or sequence regions they share, more similars are going to be those phenotypes. And great question. I don't know if I answered the question or still you have questions about that. Okay, we have more questions here in the chat. What percentage of the training population is considered from total genotype populations? So what we do Tesla, it's that basically we split the population. 70% of the population is used to create the model, and the remainder 30% of the population is using to test the model. So in that scenario, you need to genotype all the population, and basically you just split that, and you don't need the phenotype in the testing population, but you need the phenotype in the training population. But if you are using genomic selection, or if you want to use genomic selection, you need to genotype all your lines. Because basically the training population, what you intended to do, it's try to estimate the additive effect of every single molecular marker, to estimate the allele effects. And after estimate the allele effects, you can use the genotype information of other lines to predict their phenotype. So you need the genotype for that. And Bernice asks about if I impute the missing data. Yeah, and you are right, 50%, it's a lot missing data. And that, again, that depends of the person that you are talking with. I prefer to do the imputations. And again, in Common Bean, the LD, the linkage disequilibrium, it's not low. And a lot of imputation softwares use the LD to try to impute the molecular markers. And I think that depends of the population that you have. In my case, I have a big population, so my populations are around 300 that are not like that small. So the imputations usually do a good job. And what I'm trying to do always that I use imputation, it's try to use at least two different methodologies to the imputation, and then I compare what is the output of those imputations. Okay. Do you have more questions, anybody?
Yes, I have one.
Great, go ahead.
Thank you. This is for the lecture 2, population genetics. First of all, it is about the Hardy-Weinberg assumptions, and in assumption 3, my first question, assumption 3 implies that the Hardy-Weinberg equilibrium exists when we have large population. How large is large? That's my first question. My second question is, do you think it is possible or easy within a population to have all these 6 assumptions met? So if yes, please explain. If not, how then why do we use those those formulas for computing allele frequencies and genotypes of frequencies, when we know we are basing it on assumptions that are hardly met? Thank you.
That is a wonderful question. How I can pronounce your name? Batisteva? Sorry?
Sorry, say it again.
That how I can pronounce your name?
At one.
At one. Okay, at one. Thank
Yeah.
you so much.
Okay.
The thing is that you're so right. Please mute yourself. You are right. So the first question was to, yeah, how it's big, but I don't have an answer for how big should be the population. And I think that should be between thousands and that it's more about when we are talking about when we talk about population genetics, usually we have to think about like nature population genetics. So nature population, so they're big and there are a lot of concepts of population genetics that are useful in plant breeding, but a lot of them, yeah, basically we have another, how I can say that, we have to consider that in breeding panels, you're just gonna have a couple of hundreds genotypes, and there are a lot of assumptions, as you said, of Harding-Weinberg equilibrium, for example, or population genetics in general, that are not gonna be true. So the Harding-Weinberg equilibrium concepts are really important to understand all the concept of allelic frequencies, genotype frequencies, and how you can determine if you have subpopulations in your study. Because you have to take into account about the subpopulation, so you have to take care about that. If you have different subpopulations, in some case, some analysis, you just cannot put together all the data. So the question for the first one, so how big, how long is, yeah, what is it, how is the number to see that we have a big population? Again, I think that it will be between thousands, if it's not bigger. In plant breeding, we like a lot of assumptions in Harding-Weinberg are not true. But for example, if you are using QTL analysis, please mute yourself, that I cannot hear. But for example, a Harding-Weinberg equilibrium is useful when you are working in QTL analysis, because in QTL analysis, you have to remove the markers that are not under Harding-Weinberg equilibrium, because you have to, if one marker is not under Harding-Weinberg equilibrium, could be because errors in the genotyping methodology. So we can use Harding-Weinberg there in QTL. In GWAS and in genomic selection, we don't use like a Harding-Weinberg. In GWAS, we use that because we have to identify if we have subpopulations, and if we have subpopulation, we have to take into account that in the model as a fixed variable, because if we don't, we're going to have different allele frequencies in both subpopulations that we're going to put together. That is something that we're going to discuss next week. And if we put together two populations that have different allele frequencies, we're going to create a false LD between markers and QTLs. So that is going to be a problem. And in genomic selection, we have the same problem. So if we have two subpopulations and we create a model with one population that have one allele frequencies, and we try to predict that another subpopulation with different allele frequencies, that is going to be an issue as well. So the answer is that the Harding-Weinberg equilibrium are not true in breeding programs, but in some degree we have to take into account about them. Okay. And it's 9. Okay, for you it's 4 p.m. If you have additional questions, this is the moment to do them. And if you don't, yeah.
Excuse me, Poro?
Right.
Yes, this is Symphony. I just asked a question. I don't know whether you have been able to read it about the testing of Adi-Weinberg equilibrium, where you put a formula on chi-square. I read that when the degree of freedom is around 1, there is a correction factor of 0.5 that should be applied within that formula. I don't know whether you have been able to find that correction factor in your reading. Thank you.
Could you repeat your question, please?
Yeah, when we are testing the population for Adi-Weinberg equilibrium, we use a chi-square test. And when the degree of freedom is around 1, there is a correction factor of 0.5 that should be applied. I don't know whether you have found it somewhere in your reading, because in your slide you only apply observed frequency minus expected frequency without applying the correction factor of 0.5. That's what I'm asking. Thank you.
Oh, I don't know. I don't know. Like, yeah, I don't have an answer for that. I haven't read about that. Yeah, but as far as I know, or the way that we can check for with the chi-square formula, it's with 1 degrees of freedom, because as far as I know, as we have 3 genotypes, we have to take into account first one, take 1 or subtract 1 because the finite number of the population, that is kind of a standard, and another one because we have to estimate the allele frequency of E, and that is why in that slide I use 1 degree of freedom. But yeah, maybe you are right. But yeah, I haven't read about that.
Yeah, in fact, the degree of freedom is the number of genotype minus the number of alleles. So in case you have more than 1, that formula cannot be applied.
Oh, okay. So that is why I said at the beginning that I'm not an expert. So thank you so much for your comment. And that is good. And that is why the Slack channel also is good, because if you have comments, if you have thoughts to share, that it's a good place to put your questions and concerns.
Thank you.
Okay. Any another question or comment? Okay, so again, please remember for next week, post your questions. Post your questions 2 days before. That will be good, because in that way I can prepare a little better the answer for each question, and remember that it's like an space to share what you think, your questions and your comments. So don't be shy. You can open your microphone and to ask questions or to share your thoughts about the course. And again, this is the first course that or workshop that I'm trying to do for myself. So yeah, please be kind with me. And if you have questions or if you have some feedback, yeah, I'm happy to receive that. Okay, and if you have not more comments, do you have another question? No. Okay, if you don't, we can finish now. And I'm gonna post the video for the second week in 1 hour. So I have to post that in the website. And please join to the Slack channel. It's really important for the course. And I think that for you, if you want to connect with other people that is interested in quantitative genetic. Okay, everybody, thank you for being here, and see you hopefully next week.
Okay, thank you so much.
Okay.
Thank you so much.
Thank you, Paulo.
Thank you.
Thank you, Paul.
Thank you. Bye, everyone.
Thank you.
Bye.
Bye.
Thank you so much. Bye.
Thank you.